It is clear that today's speakers have drawn quite the crowd, and I appreciate all of you attending. Today, we are really very fortunate to have two of our leading faculty who both have taken science to innovate in a way that has impacted the care of patients across the globe. And I think these are really shining examples of the many members of our faculty who really do move science into the clinic. So I will start with our first speaker, and I will say both really need no introduction, but our first speaker is Dr. Lieping Chen. And Lieping, as you know, is the United Technologies Corporation professor in cancer research and a professor of immunology, dermatology, and medicine, and is also a co-director of our cancer immunology program at the Yale Cancer Center. Lieping's achievements in cancer immunology are legion, and obviously his work in understanding PD-1 and PD-L1 was instrumental in actually changing the landscape of cancer care probably for generations to come. And so it really is my distinct privilege to introduce Dr. Lieping Chen.
Thank you, Charlie, for introduction. So today, I'm going to briefly, in beginning, just briefly introduce what the concept of cancer immunotherapy is. And I think it's this because of this, the anti-PD-1, PD-L1 therapy is now actually make us think the quite different directions now for the cancer immunotherapy. So this title, you can see the end of beginning is because we started new thinking now about this field. Then later, I'll show you some data. But we'll be very brief. Talk about what we are doing. Okay, what we think is can bring this cancer immunotherapy to next level. So this is a disclosure. So the traditional thinking for the cancer immunotherapy is like this: it's a car racing game. Okay, is you have tumor growing in the body, then you have immunity developed accordingly. Then this started racing. Okay, tumors start growing, the immunity start develop. Then if the cancer grow faster and the immunity cannot catch up, then cancer beat immunity. Okay, other way around. Then if this cancer, if the immunity can catch up or even stronger than the cancer, then the immunity will beat cancer. Okay, so this is very traditional thinking. So based on this thinking, then because this influences the strategy of how you develop cancer immunotherapy. Okay, so I'm going to illustrate a little bit later. So the now current view is very different. We know the cancer not just keep growing faster and faster. Is cancer also try to stop the immunity? Okay, we call immune evasion mechanism. Okay, cancer develop lots of mechanisms try to stop the immunity. Okay, so it's not really simple, just race game. Is the really cancer can start counterattack immunity and then try to hold the immunity to fail. So because of these two different thinking, the two types of thinking, then will lead to a very different approach for cancer immunotherapy. Okay, so first type, which is based on this enhancement ideas, is you have since we talk about here is the immunity, intrinsic immunity. It may be not sufficient, okay, because the tumor grow faster, right? So that's the original idea. Then the immunity is not sufficient or not able to catch up. Then we need to enhance the immunity, okay? So all the normal immune mechanism is under study, and the basic idea is to amplify them, okay? Amplify them 10 times, 100 times, or 1,000 times better, and see this immunity can control the tumor, okay? So now we have different ideas, which is called normalization, okay, which is based on this immune evasion mechanism. So which is based on belief is intrinsic immunity might be sufficient already, okay? So which means host immunity are capable of control tumors. But the problem is because of this counterattack mechanism, tumor will develop, and then they can hold the immunity, okay? So then all you need is to then strategy will be quite different. Then it's you need to identify the problem and then to restore the defect, particular mechanism. Then let immunity go back to the normal level, then it's sufficient. Okay, so you can see these are two different ideas. Okay, so so when last year I was spend some time and try to summarize or to read through larger history of the cancer immunotherapy. As larger you probably know, is the first, the typical, considered as classical immunotherapy is interferon, type 1 interferon. It's developed in '84, approved by FDA, and for the treatment of the hairy leukemias and the kidney cancer melanoma. Okay, so that considered is the first actually FDA-approved immunotherapy. And since then, there's a few others developed based on these enhancement ideas, okay, which is IL-2, and then there's later is the anti-CTLA-4, you can see, and vaccine. All these are based on enhancement. Okay, enhancement, as I mentioned, is not based on defect of immune response. It's based on how to amplify immune response. Even this is a normal mechanism that's amplified. Okay, so that's the idea. Now you can see on the right side, which is the anti-PD-1, PD-L1 therapy, okay, which is approved in 2014. Now you see how many indications, more than 10 indications in the cancer being approved, and including all the larger major, the solid tumors especially, and but there's a very limited improvement by enhancement ideas. Okay, you can see there's a huge contrast, and this take 35 years to now. Okay, and to develop few indications of cancer immunotherapy, but anti-PD-1, PD-L1 actually move much faster. So I think from here, I think the key is we need to learn something from here. Why this therapy is much more effective and much broader indication, and also much less toxicities. Okay, so this is something we wanted to learn. So this is the cartoon actually showing the kind of major differences of these two types of therapies. Let's consider immune response as the water flows through the pipe. Then when tumors start growing, then you have block the pipe. So there's two strategies. One is enhancement, which is then literally increase the pressure of the water. Okay, force the water to get through. Okay, so by that way, consider this is the enhancement. Okay, but normalization is different. Normalization is try to find where the block is and selectively remove the block. Okay, that's through. So you can see the differences then is with the enhancement, you might see some side effect. Okay, you might have a lot of problem because you try to force this pressure obviously increase, and then you do have some trouble. But block, then the issue then is you have to find the block. Okay, so it's not always easy to find that. So the enhancement immunotherapy is based on the general idea is that you have, we know this is a fundamental cancer immunity, how this cancer immunity can generate. When you have a tumor growing in the body, then they will have lots of energy. They will make a lot of energy available to the immune system. Okay, this energy could be mutated proteins, could be the amplified, overexpressed proteins. They're all available to a subset of cell called antigen presenting cells. These cells are either dendritic cells, macrophage, or B cells. They can then process the antigen and then migrate and then bring this antigen to the lymphoid organ. Okay, in the lymphoid organ, they will meet the lymphocyte. Okay, these lymphocytes then will activate lymphocyte. These activated lymphocytes will come out, become effector cells. Okay, we call effector cells. The effector cell finally will come back to the tumor and recognize that particular antigen and lyse them. So this is a general process. Now, enhancement immunotherapy is basically designed based on this finding, based on this basic process. It's the cancer vaccine, which is based on identify antigen, okay, and then make antigen presenting cell present better, make them migrate better. Okay, so then another strategy is when T cells, in the lymphoid organ, try to increase the amplification. Okay, try to, let's say, co-stimulation or anti-CTO4, which is deplete the T regulatory cells, then it will make T cells proliferate better. Cytokines, okay, can promote survival, can promote proliferation. And adaptive cell therapy, that's another strategy, which is very straightforward. Can isolate this effector cell in the blood or from the tumor site, okay, and in vitro educate them, okay, amplify them, may engineer them to make them a lot better, okay, cells, and then infuse back directly back to the blood, okay. So hopefully they will go back to the tumor. So all these strategies. In fact, if you go back to look at the history, all this strategy is immunologically successful, okay? Meaning every strategy actually lead to the appearance of better T cells in the blood, okay? Either by vaccine or the co-stimulation, cytokine, CTO4, or the adaptive transfer, okay? You get, meaning is immunologically successful, meaning is when you give a vaccine, for example, you can get right T cells, okay, which recognize the vaccine, recognize a particular antigen. And adaptive therapy, all this lead to good T cells. Unfortunately, this is the amplified or enhanced T cell immunity is not always lead to clinical regression of tumor. Okay, this is one of the dilemma has been in cancer immunotherapy field for a very long time. Okay, meaning is better immune response not always lead to the clinical response. So then the field in the about 2000 have a major crisis. Okay, is the people believe the about 50% of people, or maybe even more, okay, believe say cancer immunology or immunity is not going to work because you show clearly there is cancer immunity in the blood, but tumor is not regress. So idea is then is very clear. It says immunity is not going to control tumor. Okay, so those people, most of them walk away, leave the field. Okay, but then there's still have some people still stay in the field which believe there were different things. Like what our lab actually believe there is some problems in the tumor microenvironment here. Okay, even you have all these processes are successful, eventually you get a good effector T cells. But those T cells, when come to the tumor site, try to get rid of the tumor, they get in trouble. Okay, it's because tumor develop all this evasion mechanism, okay, which selectively get rid of effector cells. So final step, killer step of the immune cells will not be able to execute. Okay, so this is the general idea. We have actually tested this idea, spent lots of time to work on that. And so this is just summary of the enhancement cancer immunotherapy. So this is not, this is actually a huge effort. Okay, you can see cytokines, you know, there's a lot of negative trials. Okay, there's a positive trial, as I mentioned, but lots of negative trials. It's not like we didn't try hard. Okay, we tried very hard actually. You know, cancer vaccine, which is, you know, a couple thousand trials, okay, which is all failed. Actually, negative trials in the majority of solid tumors didn't work. Okay. Anti-CTO4 is not like a try, not high enough. Okay, more than 250 trials being tested, largely because of toxicities. We know cell therapy right now there's successful in the hematopoietic cancers, but not in the solid tumors. Okay, so all this, in fact, has been tried pretty hard actually, but limited success. So why this majority of trials fail? One is because of toxicities. Okay. Lots of those agents, okay, before you see the effect, toxicity will not let you continue. Or low efficacy, we know there's a vaccine, in many cases is not have sufficient efficacy, or CAR T cells, which is not in the solid tumors. So we'll skip this one. And now you know there's a concept called immune checkpoint blockade, okay, which is pretty misleading, I should say. Based on that concept actually is literally any inhibitors, okay, you can make an agent against that and then try to remove that. And then we see that frequently those trials fail, okay, we continue hearing all these trials fail. So it's not just simple checkpoint, okay, can do the job, okay? So this is what the lesson we learned. So just tell you briefly what we have done in the past. So we have in 99, we actually in that time we discovered one molecule in the tumor microenvironment. Okay, we specifically looking at something highly expressed in the tumor microenvironment, but not in the other part, because we believe there's a microenvironment have a problem. Okay, as I mentioned a while ago. So the first story was 99. We cloned this gene B7H1. Okay, and later this molecule being called PDL1. Okay, and because the Tatsuku Hangzhou, as you cloned this PD1 long time ago, and then they don't know these two actually molecules go with by parallel. They don't really know the function is. So then in 2000, after the cloning, then they found this molecule bind each other. Okay. So in this stage is basic science. Okay, we just identify a pathway which is potentially immune inhibitory, but that's not enough. Okay, if you don't move to the cancer therapies, that's not enough. So we actually work on that and then found this molecule is highly induced in the tumor microenvironment only. Okay, minimal in other tissues, but do have other tissues because the other tissue, if there's a subclinical the patient, sometimes they do have upregulated PD-L1, which might be responsible for the, right now we see some autoimmune reactivities. And we found this molecule can inhibit tumor immunity, and then we actually make an antibody. And then in that time show in the animal study, they can block this interaction, then can lead to increased immune response in the tumor microenvironment. So this is one of the early experiment principles. So encouraged by this study, we actually then convinced the company, okay, called Matrix, and then moved this humanized antibody to clinical trial. So the first clinical trial when I was at Johns Hopkins started 2006, and then lead to finally lead to this antibody finally go to clinic. So there's a lot of different mechanisms being described. We actually describe a couple of potential mechanisms of how PD-1 and PD-L1 interaction, how they inhibit immune response. Okay, like say, T cell will release IL-10, it will induce apoptosis, T cell energy, all this being published. Okay. So then the next, after the discovery of this interaction, then take us literally 10 years to figure out how it works. Okay, now I just summarize what we describe now. Okay, is what we know about how it works. We describe this particular pathway to inhibit immune response by when you have these effector T cells, as I mentioned, come to finally when they already fully activate, when they come to tumor site, they will recognize tumor antigen, right? Then after recognize tumor antigen, they will start attack those tumor cells. Now there's a couple of things happen. One is this T cell then will release a cytokine called interferon gamma. Okay? We know interferon gamma is a very good thing. They can help in MHC class 1 upregulation, T cell survivals. But unfortunately, interferon gamma did a very bad thing, which is the major cytokine right now, okay, we know is they can upregulate PD-L1 on the tumor site, okay? And literally in any tumor cell, any cells actually in the tumor microenvironment. So this PD-L1 then binds to PD-1, which is majority of effector cells already have, okay? Binds to that and then lead to the death of the T cells. So now you can see this mechanism works in the final stage of the immune response to tumor. Okay, when everything is ready, make all the effector cells finally come to tumor site and get shut down, get paralyzed in the end. Okay, so this is how it works. So as you all know, there's now 6 drugs being approved by FDA. Okay, already all in clinic. So to summarize, what we think is this uniqueness of this normalization cancer immunotherapy is, first, you have to identify the immune evasion mechanism. Okay, what particularly problem there, and then to restore this defect. Okay, this is how this anti-PD-1, PD-L1 worked. Number 2 is this immune evasion happened in the tumor microenvironment, but not systemically. Okay, because now when we go back to study patient, you can see the immune response in general is pretty healthy, actually in the other part of the body. but not in the tumor site. Okay, so this is the major problem site. And the obvious also another third things, which is we need to identify a master switch, which then could restore or reprogram entire immunity in the tumor microenvironment. This is becoming critical because lots of those so-called checkpoints, they are capable of block particular defect or particular, the step of immune response, but they will not be able to reprogram entire microenvironment. Okay, they can hit that particular pathway, but that pathway is not big enough or not master enough, or there's a multi-pathway there. Okay, this one pathway might not sufficient to reverse change entire microenvironment. So that is become an issue. So we basically use these three criteria to identify our new the immunotherapy agents. So I will show you a little bit later. So we then how we approach this thing. Okay, we know this after get all these ideas, we then just based on these two things. One is the T cell infiltration. We know that's provide a PD-1, right? And it's very critical for the killing of tumor cells. And then there's expression of PD-L1. Okay? Based on these two simple markers, when you look at the patient, get into patient, okay, this is a lung cancer patient. Then you can clearly see there's 4 types of tumors. Okay, so we classify this just very simple classification. Okay, based on this type 1, okay, missing of T cells and the PD-L1. Type 2 would be double positive. Okay, both is there, right? And the 3 and 4 is only have 1. Okay, 3 have T cell only, but not PD-L1. And then type 4 is only have PD-L1, but not T cells. So now by look at this, then you almost know immediately says PD-1 or PD-L1 blockade therapy would be only benefit for this type of patient, type 2, okay? Because the mechanism is there, PD-1 is there, PD-L1 is there. They are interacting and inhibit immune response. Now you give antibody, you block that, okay? So type 2 patient will work, which is actually now turned out to be true in the clinic. And all type of 3, or other type will not work. Why? Because they simply missing target, okay? So we actually call it target missing resistant, okay? You can say it is resistant because I know clinicians like to say if drug don't work, you say it's resistant. So all other is resistant, going to be resistant, but we call it target missing resistant because there's no target there, right? So cartoon will be shown like this, okay? You have T cells here, you have tumor cell here, right? You have T cells, and then this is type 1, 2, 3, 4. So when we put a large data together, okay, this is a collecting of all these the multi-study, including ours. And you put all different type of cancer here, melanoma, let's say 256 cases, lung, a couple hundred. And if you put them into these 4 different categories, okay, then it's quite interesting. The number come up quite interesting. So this is total 2,000, okay. And we see type 1, which is double negative, which is 37%. Double positive, 24%. This actually match what we see in clinic right now, responding patient. Patient response to the therapy, okay? This is the average responding rate. Obviously, it goes to different type is a little bit different, okay? Type 3, which only T cells but PD-L1 is not there. That means these T cells are not functional because they do not release interferon gamma, okay? Which is another quarter, okay? Then there's type 4 is about 13%. So overall, this is our strategy, okay? We believe right now this therapy is only work in this type 2, okay? Which is now large data is coming to support that, okay? Which is about quarter of the patient in general, okay? Maybe different type of patient, different type of diseases is a little bit different, but overall it's about 25%. So then there is type 1 and 4, which is the no T cells, okay? So the problem, this is about 50%, okay, a large portion. And this type of patient, the first problem is T cell is never come to the tumor. T cell ignore this tumor, okay? So literally means there's no immune response against tumor, okay? So the problem is different. We are actually active work on this. And first try to figure out why T cell is ignoring tumors. Okay, why is that? Okay, so that's the first fundamental question we ask. And now type 3, which is about a quarter patient, okay, which is you have tumor, you have T cells coming, they actually identify the problems. They know there's a tumor there, but there's no interferon gamma release, no PD-L1, okay? So there's other pathway there, right? Most likely is other pathway is there operating, okay, independent of the PD-L1. So this is we actively also work on that. So we took the comprehensive approach. This is a very big approach, and obviously very expensive too. So we have spent now more than 10 years actually to establish two big platforms. These two platforms is one is called genome scale receptor array, okay, which is basically we have all the cell surface molecule, which could be targeted by antibody. Okay, so we have all the 8,000 genes basically in our plate. Individually, we can express them and then screen them for binding to which. So it's like PD-1 binds to PD-L1. Okay, that kind of screening system. Second platform is T cell activity array, which is we then can express each of these 8,000 molecules in a cell, in artificial cells, tumor cells or antigen-presenting cells, and to show they can inhibit T cell response. Okay, so we can do large scale screening to show those inhibitors. So to identify the inhibitor is just first step. Okay, second step then is we need to put this in the context of which type of tumor. Okay, as I mentioned, there's 4 different types. So then we spot them to the individual type of cancers. Okay, then after that, then we study the function, okay, to see they really play a role in the animal model and in the human. And also the expression profiles. As I mentioned, they has to be specifically upregulated during the tumor progression in the tumor site. Okay, we don't want it to express. We don't want to work on those molecules expressed all over the place because that will lead to trouble. Okay, with the toxicity basically. So by that way, I'll just summarize here. I don't think I have a date. I have a time to show the data. As you can see, the type 2, which is double positive, we have PD-1, PD-L1 already, right? Can attack this type of tumors. Recently, we also found another molecule called FGL1. This molecule binds to LAG3. Okay, they can express on either type 2 or type 3 tumors. And also another one is called Siglec-15, which is exclusively expressed in the type 3. Okay. Meaning is they don't express in here. Okay, we still work on others, but others are unlikely because there's no T cells. So the mechanism of this molecule is binds to a receptor on T cells and inhibit immune response, independent of the PD-1, PD-L1. Okay, and also as you know, type 3, there's no PD-L1 expression, but express Siglec-15. So this is kind of interesting. It's Siglec-15, and whatever you have a tumor express Siglec-15, they are most unlikely express PD-L1. Okay, so this kind of molecule. So now we know why. Okay, it's because they have different mechanism. Like for example, interferon will shut down. Interferon will upregulate PD-L1, but will shut down this molecule expression. Okay, so this is one reason. So we already put this in the trial. Okay, the phase 1 clinical trial already started. So we have now. So we talk about I believe we have large tools can deal with type 2 and type 3 patient. And now we go back to work on type 1 and type 4. Okay, hopefully next time I will report to you what our finding is. So to summarize what I talk is there's a 2 type of ideas in the immunotherapy, 2 approaches, and we just started to understand that. Okay, and then to put them in practice. One is enhancement, which is tradition and has been a long time. Okay, it's basically amplify tumor immunity, which could be well above the physiological level. So that's why you can start to see a lot of toxicities. There's a limitation there, and but an advantage is the target for this approach is unlimited. Okay? You basically can spot any molecules in the process of immune response. You just amplify them 100 times higher, or 1,000 times higher. Okay, you got one type of new immune therapy. Okay. And so this approach still continue, still very popular, okay, in the field. But normalization is the newer one, which is based on understanding of what is in the tumor microenvironment, what's wrong is there, and how can bring them to the normal level. Okay, so obviously the disadvantage is many targets still need to be discovered. Okay, and the need to be evaluated individually in the clinic. So that will take time. Okay, so at Yale, we actually work very closely with a group of physicians, scientists, and Roy Herbst sit there. we work on lots of lung cancer through NCI lung support, NCI skin support, which is the Mario Snow and Marcus Bosenberg's pathologist. Obviously, is very critical for us. The David Rims and then Kurt Schalper are very close collaborators. Anthony Kim is a thoracic surgeon. We work with him for quite a few years to analyze the sample of the study. So my lab, Yale, Jun Wang, the Sang Jing Wei is a grad student, Jun Wang is a fellow, and the Akedi is a pathologist, and Tina is the graduate students. And then, as I mentioned, the clinical trial, Secret 15, we already bring to clinical trial, work with a company called NextCure, and now it's already ongoing phase 1. We have also lots of collaborators. Spain is ongoing trials, so we can get a lot of sample for analysis. Okay, thank you.